NGS stands for Next Generation Sequencing, also known as High Throughput Sequencing. It refers to the set of modern sequencing technology that allow for rapid and cost-effective determination of sequences of DNA and RNA. NGS has revolutionized genomic research by enabling the sequencing of large amount of genetic material in parallel. NGS technique differ from traditional Sanger technology, which was the dominant method prior to the advent of NGS. The key advantage of NGS is its ability to generate a massive amount of sequence data in relatively short time and at a low cost per base pair. This has greatly facilitated various genomic applications, including genome sequencing, RNA sequencing, chromatin immunoprecipitation sequencing, DNA methylation analysis, along with metagenomics and exome sequencing. Common platforms for NGS include Illumina, Iron Tolerant, and Pacific Biosciences, each with its own technology and workflow. The widespread adoption of NGS has played a crucial role in advancing fields such as personalized medicine, cancer research, and understanding the genetic basis of various diseases.